Hello students, my name is Dr. P. Shashikiran. I am working as lecturer in botany at SKP Government Degree College, Guntakal. So today's topic that I will be dealing with is cells, types of cells and differences between a eukaryotic cell and an prokaryotic cell. So the objectives of today's class is what do you mean by a cell, then brief history of cell, then types of cells, then the major differences between a prokaryotic cell and an eukaryotic cell. So what do you mean by a cell? How can you define a cell? So cell can be defined as a structural and a functional unit of any living organism or body. So for the first time, it was Robert Hooke, the scientist who observed these cells for the first time in the year 1665. He observed the dead cells under his microscope obtained from the slides of a cork plant, which is also known as quercus. And here you can see in this picture the compartment-like structures, which are the dead cells, which was first observed by Robert Hooke in in a microscope which was prepared by himself. Later on, another scientist, Anton van Leeuwenhoek, he collected different samples from different places, from animal body, plant body, from water, earth, and so on, soil, and so on. And he observed the living cells, so different types of living cells in the microscope. Then Robert Brown was the scientist who for the first time in the year 1831 observed the nucleus, one of the major components that is present inside a cell. Then as the uh, as the progress was made in the discovery of cell, a lot of observations were made, and later on the two scientists, one was M.J. Sheldon in the year 1838 and Theodor Schwann in the year 1839. These two scientists, they proposed the cell theory. Later on, another scientist, Rudolf Virchow, a German scientist in the year 1855, he also joined Sheldon and Schwann. They proposed the modern cell theory. So the modern cell theory, what it demonstrates is all first is all living organisms are made up of either cell or cells. It may be made up of either single cell or multiple cells. It may be a unicellular or multicellular. Then all the functions are the product or the functions of cell. So whatever functions are carried out in the cells, a product byproduct is produced. Then all the cells arise from the pre-existing cells, which was famously known from Rudolf Virchow. He said omnis cellula a cellulae. That means all the cells, new cells arise from the pre-existing cells. Later on, it was Fleming in the year 1882. He described the somatic cell division and applied the term mitosis to the process. Later on, Strasburger in the year 1882 described mitosis in plant cells and introduced the term cytoplasm and nucleoplasm. So if you look at the types of cells, the variation is the smallest cell, you can say it is PPLO. The full form of PPLO is Pleuropneumonia-like organism or Mycoplasma. The size is in the range of zero point 0.1 micrometers to zero point 0.3 micrometers, which is very much smaller than a bacteria. Whereas the largest cell, we can say it is a ostrich egg. So the smallest one is PPLO and the largest one is the ostrich egg. Coming down to the factors that are responsible for the size of a cell, there are three factors which are responsible for the size of any cell. So first one is the nucleocytoplasmic ratio. Another one is the surface area to volume ratio. And third one is the rate of metabolism of a cell. So these three factors, they are responsible for the size of the cell. Here in this picture you can see in the left side, these are few cells that you can find in animal body, and here you can see different types of shapes of cell from a plant cell which are found in different tissues. So here there are, you can see a chlamydomonas, cartilage cells, a bacterial cell, then smooth muscle fiber cells, striated muscle fiber cells, neurocells, smooth muscle cell, fat cell. So all are not in a same shape, but there is a huge variation depending on the uh, type of tissue or depending on the type of organ in which these are present. Same way here also in plants, we have parenchyma tissue, parenchyma cells, collenchyma cells, sclerenchyma cells, chlorenchymatous tissue cells, stone cells, and so on. So all these cells are having different shapes which are collectively forming different tissues in different parts of a plant. So this here you can see the lot of variation in different types of in different shapes of a cell. Coming down to the types of cell, you can say there are two types of cell. First one is the prokaryotic cell and second one is the eukaryotic cell. Pro, the word you can say it is earlier or primitive, and the word karyon means nucleus. So the examples for the prokaryotic cell are bacteria, monera, and so on. Then the eukaryotic cell means the word term eu means true or advanced. At the same time, karyon word means nucleus. The examples for eukaryotic cell include the plant cell, animal cell, fungal cell, protists, and so on. Here in this picture, you can see a prokaryotic cell of a bacteria cell structure. So you don't find, the major thing is you don't find a well-defined nucleus or other cell organelles. The only components that are present are majorly cytoplasm, few ribosomes are present, then the cell wall is present, and capsule which is outer than which surrounds the cell wall. The genetic material is in the present in the form of a nucleoid, which is naked DNA, which is not surrounded by a nuclear envelope, nucleoplasm, and so on. So the genetic material, that is the DNA, is naked, which is termed as nucleoid, and the plasma membrane is present, as well as we find a flagella also. So this is a basic structure of a bacterial cell, which is an example for prokaryotic cell. Same way here you can see an example for eukaryotic cell, a plant cell structure. Here you can mainly see that a well-defined nucleus is there, having a nuclear membrane, nucleoplasm, then the chromatin material is also present and the nucleolus is present. Along with the nucleus, you find so many membrane-bounded cell organelles inside the plant cell. The outermost is the cell wall, then comes the cell membrane, and afterwards there are so many membrane bound cell organelles that are spread around in the cytoplasm of a plant cell. So those include mainly a vacuole. So a plant cell has a large vacuole present inside them. Then we have endoplasmic reticulum, smooth endoplasmic and rough endoplasmic reticulums. We have ribosomes, we have Golgi bodies, mitochondria, chloroplast, then we have amyloplast and so on. So these are the few cell organelles that are present inside a plant cell, which is an example of a eukaryotic cell. So here in this picture you can see side by side the how a prokaryotic cell looks like and how an eukaryotic cell looks like. So if you just see roughly, you can say that prokaryotic cell structure is a very simple structure and with very less few cell organelles, whereas eukaryotic cell is an advanced structure having an advanced and complex structure having so many cell organelles inside them. Coming down to few differences between a prokaryotic cell and eukaryotic cell. So the first one is the organism. The examples for a prokaryotic cell are bacteria, cyanobacteria, and so on. Whereas for a eukaryotic cell, the examples are protists, fungi, plant, and animal cells. Second one is the cell size. If you look at the cell size, it is structure is it is a simple structure in prokaryotic cell and it's a small when compared to an eukaryotic cell and it is in the range of between 1 to 10 micrometers, whereas the cell structure of an eukaryotic cell it is a complex structure. It is large when compared to a prokaryotic cell and the size is in the range of between 10 to 100 micrometers. Then if you look at the multicellular forms, so if you cells we this multicellular form are rarely seen. Mostly these are unicellular organisms, prokaryotic cells, whereas eukaryotic cells, this is a common multicellular form are common with extensive tissue formation. So there are various multicellular forms having different types of tissues in the form of different types of tissues. Then coming down to the cell wall of a prokaryotic cell, it is made up of mucopeptides, which is rigid in nature, and the cell wall it prevents the rupture of the prokaryotic cell. Here you can see that in eukaryotic cell, the rigid cell wall is absent in vertebrates, but the cellulosic cell wall is present in plant cells. Majorly, the plant cell wall is made up of cellulose. Another difference is major difference is that is the nucleus. So this is the main factor where you can observe. If you see a slide and you can say that either it is a prokaryotic cell or a eukaryotic cell. So if at all, if there is a presence of only nucleaid without any nucleoplasm or nuclear envelope or chromatin material or nucleolus, so all these things are absent, then you can say that it is a prokaryotic cell. Whereas if you find a well-defined nucleus along with the nuclear envelope, nucleoplasm, chromatin material, and nucleolus, then you can say it is an eukaryotic cell. Coming down to the genetic material, consists of a single circular or endless double helical molecule of DNA in prokaryotic cell, whereas eukaryotic cell it has presence of many chromosomes in the form of either linear or open-ended or double helical molecules of DNA are present. If you look at the cell division, in prokaryotic cell, the asexual reproduction occurs in the form of a binary fission, whereas the sexual reproduction is observed in eukaryotic cells in either mitosis or meiosis methods. Another one is the nucleoli and the mitotic apparatus. So this includes the spindle formation, nucleoli, and the mitotic apparatus means the spindle fiber formation, which is completely absent in prokaryotic cell, whereas these are present in eukaryotic cell. mainly at the during the metaphase of the metaphase phase stage, where you can easily trace out the spindle fibers attached to the chromosomes. So all these things are absent here in prokaryotic cell, whereas these are present in an eukaryotic cell. And coming down to membrane-bounded cell organelles, as I said earlier, this is the another identification factor if you can for differentiating a prokaryotic cell or an eukaryotic cell, where prokaryotic cell won't have any membrane-bounded cell organelles. All are absent here, whereas most of them are present in an eukaryotic cell. If you see few examples of membrane-bounded cell organelles, first one is the plastid. You don't find in prokaryotic cell. Well, you find plastids in an eukaryotic cell. Coming down to another organelle, that is chloroplast, which is not found in prokaryotic cells. These are present in eukaryotic cells. Another one is endoplasmic reticulum. That's also not present in prokaryotic cell. Well, they are observed in eukaryotic cells. There are two types of endoplasmic reticulum. One is the smooth walled endoplasmic reticulum, which lacks the ribosomes, whereas the another one is rough endoplasmic reticulum, which has ribosomes attached to its membranes. Another one is the ribosomes. The smaller ones, that is the 70s type of ribosomes, are dispersed in the cytoplasm, whereas the larger ones, that is 80s type of ribosomes, are present in the cytoplasm, as well as they are attached on the surface of the endoplasmic reticulum. That's why here these are termed as rough endoplasmic reticulum in case of eukaryotic cells. Another one is the Golgi apparatus. This is also a membrane-bounded organelle similar to endoplasmic reticulum, which are absent in prokaryotic cells while they are present in eukaryotic cells. At the same time, another very important cell organelle, that is the lysosomes, which are also known as the suicidal bags of a cell. These are absent here in prokaryotes, whereas these are present in eukaryotes. Another one is the mitochondria, which is absent here in prokaryotes and present in eukaryotes. Another one is vacuoles, where these are completely absent in prokaryotes, whereas these are present in eukaryotes. And that too, in animal cells, these are either absent completely or present in a very, very small size, whereas you can find a well-defined big, large vacuole in plant cells. Regarding the photosynthetic pigments, these are scattered in few photosynthetic, there is prokaryotic cells, and there are few examples in the lamellae, whereas the photosynthetic pigments are present in the plastids, especially the chloroplasts having the chlorophylls. Then the respiratory enzymes, these are located at the on the plasma membrane of a prokaryotic cell, whereas these are enclosed in mitochondria. Then the DNA replication, here the replicates entire genome at once in case of prokaryotes, whereas a well-regulated high with selective origin and sequences are found in eukaryotic cell. Then the growth rate per generation time, if you see, it's very at a very fast rate in prokaryotic cells, whereas it is very slow pace of growth is seen in eukaryotic cell. Another one is the plasmids, the presence of another DNA fragment or that is commonly found in prokaryotic cells, whereas these are completely absent in eukaryotic cells or rarely found. Another structure, that is the flagella. These are small in prokaryotic cells, whereas we find a large flagella in few examples of eukaryotic cells. So these were the few differences between the prokaryotic cell and eukaryotic cell. Coming down to the summary, we discussed about the what is cell, definition of a cell, that is the structural and functional unit of a living organism. We saw a brief history of cell, who all saw the dead cells, living cells, and who discovered nucleus and so on. Then we saw the types of cells, that is the prokaryotic cell and eukaryotic cell, and also we discussed differences between a prokaryotic cell and eukaryotic cell in detail. So that's all with regard to cell and types of cell and differences between a prokaryotic cell, which is a very fundamental topic we discussed today. Okay, thank you.